Hi everyone, this is my first video in lipids. We know that lipids are the compounds which are obtained by reaction between glycerol and fatty acids. Simply, the lipids are the esters of glycerol and fatty acid. And we know that in case of glycerol, we are having 3 OH groups in this way. So this is the structure of glycerol. So totally we are having 3 OH groups in glycerol. And among the 3 OH groups, either 1 OH or 2 or all the 3 OH groups may get to react with fatty acid molecule so as to produce the ester of it, that is nothing but lipid. But if in 3 OH groups of the glycerol, if 2 OH groups of the glycerol are esterified with fatty acid molecule, and if one of the OH group of the glycerol get esterified with phosphoric acid, that means if one of the OH group get replaced with the phosphate group, we are going to get the derivative of that phosphate, the derivative of that lipid, and we call that compound as phospholipids. And this is the content I have taken for discussion, phospholipids. So simply we can say these are the phospholipids in which one of the OH group in the glycerol molecule is replaced with phosphate group, or else you can say in some other way, phospholipids are the compounds in which among the 3 OH groups of the glycerol molecule, 2 are replaced with fatty acid molecule and 1 OH group is replaced with phosphate group. Such a compound, such a lipid is called as phospholipid. And most of the common phospholipids are phosphoacyl glycerols, or we can, it's also called as, I said, most of the phospholipids are phosphoacyl glycerols, and these phosphoacyl glycerols can also be called as phosphoglycerides. This is the other name we can give for phosphoacyl glycerols. We can also call them as phosphoglyceride. That is, as we said, in case of phospholipid, one of the OH group of the glycerol molecule is get replaced with phosphate group. So we can write the structure of that phospholipid, the skeletal structure of phospholipid in this way. CH2O, here there will be the fatty acid molecule, and one OH group of the glycerol molecule will be get replaced with phosphate group, this one, right? And here you can observe this phospholipid molecule is having both non-polar hydrocarbon part as well as polar part. When you are having the fatty acid molecules over here, anyway you will be having the polar part as well as non-polar hydrocarbon part. Here also we are having non-polar hydrocarbon part, and once again the phosphate group is polar group. So the phospholipid molecule is having both polar as well as non-polar. We know that polar heads are always hydrophilic in nature and non-polar components are always hydrophobic in nature. And since in a phospholipid molecule we are having both hydrophilic as well as hydrophobic component, it will show both tendency towards water molecule as well as repulsion towards the water molecule. Because of this reason, we can say the phospholipid molecules are amphipathic molecules. Amphipathic molecules. These are called amphipathic molecules. That means consisting of both hydrophilic as well as hydrophobic components. In the phospholipid, we are having both polar and non-polar components, right? So including the phosphate component, in phospholipid molecule, the polar heads which are present will always prefer to exist in aqueous environment, or prefer to attract towards an aqueous environment, and the non-polar components will excludes the aqueous environment. That we can confirm actually when a phospholipid is dispersed in water. When the phospholipid is dispersed in water, the phospholipid molecule readily forms micellar. They can readily form a simple lipid monolayer, or they can form lipid bilayer, or they will form the spherical components which are also called as vesicles. And these kinds of things will be formed only when the component is having the polar group. So that due to the formation of micelles, monolayers, bilayers and vesicles by a phospholipid molecule, we can say that in phospholipid molecule there is a presence of polar heads. And among these things, we shall give a bit of discussion regarding micelles and vesicles. Okay, so first we shall consider about micellae. Okay, micellae is simply the thing is, we said in case of a lipid molecule, say, let me write in this way. So let us consider a simple phospholipid molecule, and in this phospholipid molecule, this is the polar head and this is non-polar tail. So as we said, the phospholipid molecules are amphipathic in nature. So we are having both polar and non-polar groups. So when a phospholipid molecule is brought in contact with water molecule, this polar head will always prone to face towards the water molecule, and this hydrophobic tails are forced to move away from the water molecules. So when we are having more number of such phospholipid molecules, they attract themselves so as to form a single molecule. An aggregate molecule will be formed, and such an aggregation which is formed when the phospholipid molecule comes in contact with water molecule is called as micellae. Okay, and now we shall see one such micellar formation. Here is one phospholipid molecule which consisting of a hydrophilic head and hydrophobic tail. Once it comes in contact with water, or when a phospholipid is dispersed in water, all the hydrophobic parts will force it to move away from the water, and all the hydrophilic heads will face towards the water molecule, so that they all merge together, and when the concentration of the phospholipid reaches the critical micellar concentration, it converts in the form of a spherical shape, and that results in the formation of micellae. The micelle formation by a phospholipid molecule will not occur at all the concentration. In order to form the micellae by a phospholipid molecule, there should be a minimum concentration of phospholipid molecule, and above to that particular concentration of the lipid monomer, only formation of micellar will takes place, and the concentration above to the which the formation of micellae is possible by a phospholipid molecule is called as critical micellar concentration. And that critical micellar concentration can be simply abbreviated as CMC, critical micellar concentration. So we can simply define the term critical micellar concentration as the lipid monomer concentration above to which the micellar aggregate will starts to begin in the state of equilibrium. I repeat, the lipid monomer concentration above to which the micellar aggregation will start to begin in equilibrium. That is called as critical micellar concentration. That is, the concentration required for the formation of micellar by a phospholipid molecule. And that's a bit about micellar. And now we shall go with the second thing, that is spherical components. That's nothing simply called as vesicles. Okay, so number 2, vesicles, or else let me say liposomes. Liposomes are actually the artificially prepared spherical vesicles, and they're mainly composed of a lipid bilayer. That is, liposomes are the artificial spherical vesicle. They are spherical in shape, spherical vesicle, and they mainly composed of lipid bilayer. And these liposomes are almost looks like a micelle, but there will be a more number of concentration of lipid bilayer over here. And these liposomes can readily fuse with the cell molecule, and they can readily mix with the contents of the cell in the intracellular medium. And this is more helpful in treating the diseases actually. That is, liposomes can be, liposomes can be made as a target molecules, and they will be developed to selected populations. Means, in order to cure a particular type of disease, if we are supposed to send a drug intravenously, so then if that drug is not supposed to dissolve in water, and if that has to act on a particular part of the body, then in such cases the drug molecule will be encapsulated within the liposome, and that liposome will be introduced into the body, and that liposome will travel through the body. And since the liposomes will be surrounded by water, and there is a lipid bilayer in liposomes that prevents the immune system in the body so as it won't undergo that much faster degradation. So whenever it goes to the particular site, there it degrades, and that releases the drug material, and that actually act against a particular type of population, and that collapses. Especially this liposomes will used as a contrast agent, especially for cancer cells. And apart from that, the liposomes are used in order to deliver the drugs, as well as the liposomes will be used in treatment of the diseases as therapeutic enzymes also. And we shall see one of such example where liposomes can be used as a materials for the releasing of drugs. Here is one liposome which consisting of aqueous solution core, which is surrounded by a hydrophobic membrane in the form of lipid bilayer, and in the core there is a drug crystallized in aqueous fluid and a DNA, and this lipid bilayer is surrounded with a protective layer which acts against immune destruction. And there are some targeting ligands on the surface of the liposome which helps in the accumulation of the pathological area for the treatment of disease. Once it accumulated, it fuse with the cell membrane and deliver the contents of it, and so that it initiates the curing of the disease. Nextly, the classification of phospholipids. Phospholipids are mainly of two types. The one type is called phosphoglycerides, and the second type is called sphingolipids. These are the two types of phospholipids. So how to differentiate the phosphoglycerides with that of sphingolipids? It's a very easiest way. In case of phosphoglycerides, the alcohol that is present is glycerol. Glycerol is the alcohol present in phosphoglycerides. Fatty acids are common and phosphates are common. But if you come to the case of sphingolipids, Here we don't have the glycerol, but instead of glycerol, at the place of alcohol, we will be having a other alcoholic content called as sphingosine. Sphingosine is the alcohol present in sphingolipids. Glycerol is the alcoholic material present in phosphoglycerides. Thus, we are calling these materials as phosphoglycerides and sphingolipids. Okay, and now we shall give the discussion regarding both phosphoglycerides as well as sphingolipids. Firstly, we shall consider about phosphoglycerides. Phosphoglycerides. These phosphoglycerides are actually covers the major portion of the phospholipids. Compared to sphingolipids, the major portion of the phospholipids were covered by phosphoglycerides only. And these phosphoglycerides are present in mainly majorly it is present in biological membrane, and these phospholipids are present in both plants as well as in animals. In both the cases, the phosphoglycerides are present, and the phosphoglycerides are more abundantly present in animal organs, such as it is mainly present in heart, as well as it is one of the main component of the brain. It is present in kidney, and it is present in the egg yolk, as well it is present in soybean also. And these are the materials wherein we can observe the presence of phosphoglycerides. If you come to the case of structure of this phosphoglycerides, structure of phosphoglycerides. Here we already said, in case of phosphoglycerides, the alcoholic material present is glycerol, and in glycerol we can see there are 3 OH groups like this. And we already said, in case of phospholipids, among the 3 OH groups, especially in case of phosphoglycerides, 2 OH groups will be replaced by fatty acid molecules, and this will be esterified with the phosphoric acid. That means phosphate group will be there. So here the first and third position both are having primary alcoholic group, and this one is a secondary alcoholic group. So when this position is esterified with phosphoric acid and this position is esterified with fatty acid molecule, though the positions are having primary alcoholic groups only, but still these positions are stereochemically not identical. The reason is, this is in attachment with the fatty acid molecule and this will be in attachment with phosphate group. So due to presence of different groups, the stereochemical similarities cannot be observed. So that in order to give the exact structure of a phosphoglyceride, in order to say the exact name of a particular phosphoglyceride, we are supposed to say the exact position at which the phosphate group is present in the glycerol molecule. And for that purpose, we will use the concept of stereospecific numbering system. Stereospecific numbering system. Just we will write the Fisher projection of this particular formula, this particular molecule, by keeping the OH group on the left hand side. So we will write in this way: CH2 OH, CHOH, CH2O, P double bond O, OH, OH, and this is the phosphorylated glycerol molecule. So here we will give 1, 2 and 3. So the phosphate group is present at third position, and we will indicate that third position as in this way: glycerol. At third position the phosphorylation is occurred, so glycerol 3-phosphate. But in order to say that third position is a stereospecific for the phosphate group, we write a prefix in front of the name as SN. This SN is called as stereospecific number. Stereospecific number. And here the stereospecific number is 3. So before giving the name of a particular phosphoglyceride, we are supposed to put the prefix SN and we are supposed to say the number 3 before writing the name of a particular phosphoglyceride. In all phosphoglycerides, we will be having this SN glycerol 3-phosphate moiety. But we said in case of phosphoglycerides, we will be having one phosphate group at third position, and the first and second positions are get esterified with fatty acid molecule. So if you have written the complete structure of this phosphoglyceride, CH2OH will get esterified with the fatty acid molecule. So our CH, and this is also esterified with fatty acid molecule, and third position, that is CH2 O, P double bond O, OH and OH. So 1, 2, 3. So third position is stereospecific for phosphate group, and we call this material as third position SN, and this whole material is simply called as phosphatidic acid. This is the complete molecular structure for 3SN phosphatidic acid. And when one of the H component among these two, one of the H component of phosphatidic acid is get replaced with a particular group, that actually results in the production of phosphoglycerides. And now we shall see some of the simple examples of phosphoglycerides, and as well as we shall see where they actually show their biological activity. Number 1 under phosphoglycerides is cephalin. Cephalin is one of the simple example of phosphoglycerides. And now we shall write the structure of phosphoglyceride, that is cephalin, CH2O, C double bond O, R1, C, H, O, C double bond O, R2, and CH2, O, P double bond O, OH, and here it will be O, CH2, CH2, NH3 plus. So here we got H atom priorly in phosphatidic acid, and that H is replaced with this particular component. So there are two carbon atoms, so ethane, and that is amine, and since there will be one OH group next to this, so it is ethanol amine, which is present at third position. So 1, 2 and 3. So we can say the complete name of the cephalin as third position, and it is the stereospecific number, and this whole component gets the name phosphatidic acid. It is a derivative of phosphatidic acid, right? So H is removed, so it will become phosphatidyl, and this component is ethanol amine, and this is the actual name of cephalin, 3-SN phosphatidyl ethanol amine. Okay, and now we shall see what is the biological importance of this cephalin, and now we shall see what is the biological importance of this cephalin. Okay, and this cephalin is actually present in all the living cells, and in human physiology this cephalin is mainly found in the white matter. It is mainly present in the white matter of that spinal cord. It is present in the white matter of the spinal cord as well as the white matter of the brain. And we know that the white matter which is present in the spinal cord as well as brain, it actually improves the speed and the transmission of the electrical nerve signals along extensions of the nerve cells, what we also call that as axons. Axons. In this we can see the white matter which is mainly composed of cephalin in brain as well as in spinal cord. This cephalin will improve the speed and the transmission of electrical nerve signals along the extensions of the nerve cells, which are simply called as axons. So that cephalin is very much useful in the proper transportation of the proper transmission of the nerve signals. Now we shall go with the second example of the phosphoglycerides. Number 2, it is lecithin. Lecithin is having the structure CH2O, C double bond O, R1. And here in case of lecithin at third position, the hydrogen of the OH group which is present at the phosphate will get replaced with N. So in case of cephalin, here we got 3 hydrogens, and here in case of lecithin, those 3 hydrogens will get replaced with methyl group with a positive charge, and this whole part is called choline. And since the choline part is replaced, the hydrogen of the phosphate group is get replaced with the choline, hence we call this as the complete name of the lecithin is 1, 2, 3. Third position, it is the stereospecific number, and it is a derivative of phosphatidic acid, so it is phosphatidyl choline as the full name of lecithin. And now we shall see what is the biological importance of this 3-SN phosphatidyl choline. And this lecithin, actually among all the phosphoglycerides, this lecithin is the major phospholipid which is present in all the living cells, means it present both in plants as well as in animals. And this lecithin is the key building block of cell membrane. It is the key building block of membrane bilayers. And in particularly, it makes up a very high proportion of outer leaflet of the plasma membrane. So in plasma membrane, it is present, this lecithin is mainly present in outer leaflet of plasma membrane. Outer leaflet of plasma membrane. And this lecithin is also present in or else we can say it is the principal phospholipid which is present in circulating fluid. That's nothing but what? Plasma. And where it is the principal component of the lipoprotein. And there are two kinds of lipoproteins actually. One is called high density lipoprotein HDL, and other one is called low density lipoprotein, that is called LDL. Simply the lipoproteins are nothing but cholesterol. And this lecithin is a very important component because in brain it this lecithin forms protective layers around the tissues. It forms protective layers for tissues in brain. So in this way it gives protection for the brain also. And now we shall go with the next example of phosphoglyceride, that is number 3, phosphatidyl serine. We know that serine is one of the amino acid. And now we shall see the structure of this phosphatidyl serine. This is the simplest structure of phosphatidyl serine. CH2O, P double bond O, OH, O, CH2, CH, and since it is amino acid, we'll be having both carboxylic group and amino group. And it is the complete structure of phosphatidyl serine. Once again, 1, 2 and 3. Number 3 is the stereospecific position, so 3SN, and it is the derivative of phosphatidic acid once again, that is phosphatidyl. The attached group is serine and is the complete name on complete structure of the material, 3SN, phosphatidyl serine. And this phosphatidyl serine is mainly present in lipid bilayer of the cell membrane. Lipid bilayer of cell membrane. And apart from occurrence in the bilayer of the cell membrane, it is compulsorily required. This phosphatidyl serine is compulsorily required for proper functioning of brain cells. So if the phosphatidyl serine got depleted in the brain, then that starts to abnormal working of the brain cells, and that may leads to many other diseases also. So phosphatidyl serine is particularly required for proper functioning of brain cells. And that's about the phosphatidyl serine. And we shall continue our discussion with the second class of phospholipids, that is sphingolipids. Sphingolipids. And we already said that sphingolipids are simply differentiated. We can easily differentiate sphingolipids from that of phosphoglycerides. That is, in case of phosphoglycerides, we are having glycerol as alcohol, and in case of sphingolipids, we are having sphingosine as the alcohol. So sphingosine is actually a long chain amino alcohol which consisting of 18 carbon atoms, and the hydrocarbon part present in sphingosine is an unsaturated part. And now we shall see the structure of the sphingosine. As I said, the sphingosine is an amino alcohol, means consisting of both amino group as well as alcoholic group, and there should be totally 18 carbon atoms. And now we shall see what is the actual structure of this sphingosine. Sphingosine is having the structure in this way: CH double bond CH 12 times, CH3 CH, and there is a presence of amine group, and once again CH2 OH. This is the simplest way of writing the structure, sphingosine. And here this sphingosine will be present in case of sphingolipid. Now we shall consider one simple example for sphingolipid. The very best example for sphingolipid is ceramide. And here we have the term cera. The meaning of cera is it is simply a wax type of material. Okay, and this ceramide is formed by the condensation of this sphingosine with a fatty acid. And here there is no reaction with OH group. Instead, the fatty acid molecule will get react with this amine group so that to form an amide linkage. And now we shall see how actually the structure of ceramide will be. So sphingosine, that is CH 12 times 1 OH group over here, CH2 OH, and here the NH2 group will get react with fatty acid molecule so as to form an amide linkage this way. So this is the complete structure of ceramide. And this ceramide is a very important material because if this ceramide is the major component present in the skin, if the depletion of the ceramide in the skins is appeared, and that finally results in the drying of skin as well as the skin will get damaged. One of the major disease that we can expose due to the depletion of the ceramide is atopic dermatitis. Atopic dermatitis is simply creation of gray and white patches on the skin, ankles, legs, and the neck, as well as other parts of the body. We shall see how the atopic dermatitis will appear. You can see in this picture the development of patches on the skin, and this is mainly due to the deficiency in the ceramide content in the skin. As well, the ceramides are very essential constituents and they act as the skin barriers actually. In humans, without having ceramides on the skin, which are acting as essential barriers, strictly speaking skin barriers, it is highly impossible to live in a dry land actually, because when we are living in a dry land where the temperature is very high, the water content present in our body will go out due to the evaporation. So if all the water content will go out of our body, then the person will suffer with dehydration. And though we are present in a very dry land with high temperature condition, instantly we don't suffer with dehydration. The main reason is presence of ceramide. The ceramide will prevent the evaporation of the water content from the human skin just by making a screen on the human skin that we can see in this picture. In the left hand part of the picture you can see that is the condition wherein the skin is lacking of ceramide. Due to lack of ceramide, the moisturous content which is present below the skin can easily come out of the skin through evaporation, and that causes the external irritation. But if you come on to the right hand part of the picture, there you can see there is a sufficient quantity of ceramide on the skin, and that avoids the outcome of the evaporation of the moisturous content from the skin, and that avoids the external irritation. So that ceramide is very much essential to avoid the external irritation on the skin.